Many of you have asked about how did AIDS start in the first place. In this video, black screen words will share the facts with you. Scientists accept that the known strains of HIV-1 are most closely related to the simian immunodeficiency viruses from wild ape living in West Central African forests. Each of the known HIV-1 strains is closely related to the SIV that infects the chimpanzees troglodytes, or related to the SIV that infects western lowland gorillas, called SIVgor. Pandemic HIV-1 strain and a rare strain found only in a few Cameroon people are clearly derived from SIVcpz strains endemic in troglodytes, chimpanzees living in Cameroon. Another very rare HIV-1 strain is clearly derived from SIVgor strains of Cameroon. Primate ancestor of HIV-1 group O, infecting 100,000 people mostly from Cameroon and also from neighbouring countries, was confirmed in 2006 to be SIVgor. Pandemic HIV-1 group M was closely related to the SIVcpz from the southeastern rainforests of Cameroon near the Sangha River. This region is where the virus was first transmitted from chimpanzees to humans. Reviews of the epidemiological evidence of early HIV-1 infection, stored blood samples, and old cases of AIDS in Central African regions have led many scientists to believe that HIV-1 group M early human center was probably not in Cameroon, but rather further south in Belgian Congo, more probably the capital city, Kinshasa. HIV-1 sequences preserved in human biological samples along with estimates of viral mutation rates. Scientists calculate that the jump from chimpanzees to humans probably happened during the late 19th or early 20th century, a time of rapid urbanization and colonization in equatorial Africa. When the zoonosis occurred is not known. Molecular dating studies suggest that HIV-1 group M had its most recent common ancestor in the early 20th century, between 1915 and 1941. A study published in 2008, analyzing viral sequences recovered from a biopsy made in Kinshasa in 1960, along with previously known sequences, suggested a common ancestor between 1873 and 1933. Genetic recombination had earlier been thought to seriously confound such phylogenetic analysis, but later work has suggested that recombination is not likely to systematically bias results. Recombination is expected to increase variance. Results of a 2008 phylogenetics study indicate that HIV evolves fairly reliably. Further research was hindered due to the primates being critically endangered. Sample analyses resulted in little data due to the rarity of experimental material. Researchers were able to hypothesize a phylogeny from the gathered data. They were also able to use the molecular clock of a specific strain of HIV to determine the initial date of transmission, which is estimated to be around 1915 and 1931. Similar research has been undertaken with SIV strains collected from several wild simian of the West African nations of Sierra Leone, Liberia, and Ivory Coast. The result phylogenetic analyses show that the virus is most closely related to the two strains of HIV-2 that spread considerably in humans are the SIVs found in the simian bays of the Thai forest in western Ivory Coast. There are 6 additional known HIV-2 groups, each having been found in just one person. They all seem to derive from independent transmissions from simian bays to humans. Groups C and D have been found in 2 people from Liberia. Group C and F have been discovered in 2 people from Sierra Leone, and groups G and H have been detected in 2 people from the Ivory Coast. HIV-2 strains are probably dead-end infections. Each of them is most closely related to SIVs strains from simian bays living in the same country where the human infection was found. Molecular dating studies suggest that both the epidemic groups started to spread among humans between 1905 and 1961. We have reached the end of the article. Please support Black Screen Words. How to support? You can do so by using Black Screen Words affiliate links for your online shopping. Link below for shopping page. You may find something you need.